Hi students, welcome back. It's Professor Bridget, and today we're going to be learning about cellular respiration. This material is found in chapter 6. And last week we learned about the type of energy that cells need to have in order to perform their daily activities. And remember, that energy is in the form of ATP, adenosine triphosphate. So, as you know, energy can never be created or destroyed, it's just converted. So, the way in which cells are able to make ATP is not kind of out of the blue, out of thin air, out of nothing. They're going to convert one form of energy already present into ATP. So, in cellular respiration, we take glucose, and oftentimes, in the presence of oxygen, that's what aerobic means. So we take glucose and oxygen inside the mitochondria, and you get ATP, and you get some byproducts as well. You get carbon dioxide, that's what is exhaled, in humans anyways. You get some water as a byproduct, and then you also get heat as a byproduct. So we're going to learn about the details of generating ATP from pre-existing energy. If we were to look at this glucose molecule, remember we were learning about potential and kinetic energy last week? And remember that molecules represent a form of potential energy. And the energy is found in the bonds of the molecule. And remember, those bonds represent electrons. Electrons are energy. So we're going to learn about how this molecule of glucose is broken down. The bonds are broken to create ATP, which is a form of energy that cells can use. So cellular respiration is used by both plant and animal cells. So they're getting energy from food in the form of glucose, and that gives or yields or produces large amounts of ATP. And as I mentioned, ATP is used to drive cellular activities, cellular work. Now, both plants and animals can use cellular respiration as a form of energy production, but plants are also unique in that they are able to use a process called photosynthesis. And photosynthesis just really means light generating, and not that it generates light, but it generates energy from light. That's this process that plants can use. They take light in the presence of CO2 and in the presence of water, and they're able to create energy. Animal cells cannot carry out photosynthesis. We'll spend some more time talking about photosynthesis in the next lecture, and maybe a little bit in the cellular respiration lecture, but mainly this lecture here is going to focus on cellular respiration. So where cellular respiration takes place is in the mitochondria. And we learned about mitochondria a couple of weeks ago, that they're an organelle inside of a cell. Now, not all steps of cellular respiration take place in the mitochondria, but the majority of the steps do. And I'll point out which steps take place in the mitochondria and which steps take place in the cytoplasm. So in this chemical reaction that we call cellular respiration, you've got a molecule of glucose. And there's a little drawing down here in the left-hand corner of a molecule of glucose, just reminding you how many hydrogens there are. Right? And remember that there's energy stored in those bonds between the hydrogens and the carbons, and the hydrogens and the oxygens. So we start out with that molecule of glucose, and in the presence of oxygen, we need to have that oxygen present in most cases. What we're really looking for is ATP, but water is a byproduct, and carbon dioxide is a byproduct. So think about, all animal cells carry out this process, but think about you as the animal carrying out this process. You've consumed some energy, let's say it's pasta or whatever, so you've consumed some carbohydrates, you breathe in oxygen, and that oxygen gets taken up by your blood and transported to every single one of your cells, and that oxygen gets sent to the mitochondria of your cells. And inside the mitochondria of your cells, you're breaking down that glucose in order to create ATP, and all of your cells use that ATP to do their work. But as a byproduct of this process, when you exhale, you get rid of some of the carbon dioxide. That's what's left of the carbons here in that glucose molecule. And then as you breathe out, the air that you breathe out is moist. There's water in it. Consider when you're wearing a mask for a long period of time. The mask gets a little wet, gets a little damp, and that's because as you exhale that CO2, some water is coming out as well, right? So even though we're kind of generalizing and saying, well, all animals carry out cellular respiration, you're an animal, and it's very relevant or much more relatable when you think about it in terms of yourself, and then kind of then carry over that understanding to all animal cells. Remember that just our general rule, our first law of thermodynamics, is that energy is never created or destroyed. So let me go ahead and erase some of the markings that I've made here. And I just want to point something out. We're going to have as many atoms on the left-hand side of the equation as we do on the right. So I want to just take account of these just so you recognize that we're not creating anything necessarily new here in this process of cellular respiration. All we're doing is rearranging, breaking bonds and rearranging atoms into different molecules. So if we think about what we start with, we're going to think about the number of carbons, the number of hydrogens, and the number of oxygens. We've lost a little bit of formatting here. This should be C6H12O6. The numbers got kind of lost there. So if we count the number of carbons, hydrogens, and oxygens on both sides of the equation, we should have equal numbers because we're not making anything new. We can only use the components that we start with. So over on the left, we've got 6 carbons. So we're going to write that in here. And over on the right-hand side, this prefix here, 6, that you see in front of the carbon, that means 6 carbons. So over on the right, you've got 6 carbons. Oops, that shouldn't be written there. We lost some formatting here too. There we go. That's where that 2 should go. So over on the right, we've got 6 carbons. Let's go back to the left. How many hydrogens are there on the left side of the equation? You can see that there are 12. So we'll write that here. How many carbons do we have on the right side? Sorry, not carbons, hydrogens on the right side of the equation. So if we look at the right side of the equation, how many hydrogens do we have? Well, we've got the 6 in front of the H, but then you've got the subscript right here. So in order to figure out how many hydrogens you have, you take that number in front of the hydrogen times the number in the subscript. So 6 times 2 is 12. So we've got 12 hydrogens over on the right. Let's go back and account for the oxygens. So if we rewrite oxygen like this, we've got 6 in front of the oxygen, but you have to multiply that by 2. So 6 times 2 is 12, right? So we've got 12 oxygens there, but also don't forget about the 6 oxygen atoms that you have here, right? So if we add 12 plus 6 together, we get 18. Now let's go over on the right side of the equation and count the oxygens on the right side. So we've got 6 times 2, right? That's 12. So we've got 12 oxygen, and then we've got to account for these oxygens here. And there are 6. So 12 plus 6 equals 18. So on the left side of the equation and on the right side of the equation, we've got the same number of carbons, hydrogens, and oxygens. The only thing that this cellular respiration process does is it rearranges atoms by breaking bonds and harvesting the energy in those bonds to phosphorylate ATP or create ATP. ATP, remember, is created by taking ADP, which is already present inside the cell, and adding a P to it, or adding a phosphate. So that makes ATP. So that energy that is released in breaking the bonds in glucose, remember, that energy goes towards adding a phosphate to ADP, adenosine diphosphate, to make adenosine triphosphate. So really, this is a chemical reaction that's just rearranging atoms to harvest energy to add a phosphate to ADP to create ATP. I know there's a lot of terminology and a lot of scientific jargon, but the more you get familiar with this, say it out loud, do the reading, write out the equation, you will get better at using these terms, and therefore you'll also get better at understanding. Okay. So let's look at some details here. Cellular respiration, as you remember from last week, is an exergonic process. Exergonic, meaning there's energy released. And this is kind of what I was just saying before. And that energy that is released is when we break down glucose, so the potential energy in glucose is used or transferred to a molecule of ATP. Cells can't use glucose directly, but they can use ATP. We get from one glucose molecule, we get about 32 ATP molecules produced. However, even though that kind of sounds like a lot, cellular respiration is only about 34% efficient, meaning there is 66% of energy still that was held in glucose. 66% of that energy is lost. So when we're converting the energy in glucose to ATP, we only really get about 34% of that total energy from glucose converted to ATP. So you might be like, well, energy is never created or destroyed. It's only transformed. Well, where does it go? Well, if we write this equation out again, C6H12O6, that's glucose. And in the presence of oxygen, you get your reactants. ATP is what we really want, right? And then we also get some CO2, and that doesn't have much energy. Can't really use that. We get some water. Again, water doesn't have much energy. But the big loss of energy here occurs in the form of heat. So heat gets lost to the environment. So that's where the rest of that energy goes. This is one of the reasons why, as humans, we're able to maintain a body temperature of 98.6. In our muscle cells, we're carrying out this process continually. We are making ATP for our muscles to use to contract. We exhale the CO2 along with some of that water, and then some of that heat is held onto by our bodies by various mechanisms. That heat is held onto by our bodies to maintain 98.6. Think about when you go and exercise and you're using your muscles are contracting and you start to feel warm. That's because in all of your muscle cells, you're converting that energy in glucose to ATP. You're placing demand on your muscles. Your muscles need ATP in order to continue exercising. So the more you carry out cellular respiration in your muscles to create ATP, the more heat you use. The more heat is lost, and your body then you're starting to get a little too hot. Then you start to sweat. Your skin feels warm, and your skin might get a little pinkish or reddish. That's because your body's trying to dump a little bit of that excess heat. But this, so I give you the example of exercising, but just all the time, this process is occurring in your muscle cells, and this is the main way that even when you're sleeping or not moving, or even when you're in a cooler environment, that we're always pretty much around 98.6 degrees. It's mainly from this process, cellular respiration, that's occurring in your muscle cells, although this process occurs in all of your living cells. Okay, so one of the important things to remember, I've said it before and I'll say it again, is that breaking bonds between the carbons and hydrogens is where the energy is released from. So in this picture, you can see some of the bonds between carbons and hydrogens. So in cellular respiration, we're going to break these bonds. Oops. I'm going to break these bonds here, right? And bonds represent electrons, and electrons represent energy. So we're going to release that energy by breaking those bonds, this process. So before we move on to some of the details, we're going to learn about two reactions. We've learned about a lot of reactions so far, such as hydrolysis and dehydration. Those were reactions that were very similar to one another. And here are two additional reactions that we're going to refer to throughout this process of learning about cellular respiration. And these two reactions are called oxidation reactions and reduction reactions. We often just put the two terms together and call that a redox reaction, reduction and oxidation. So oxidation is when one molecule loses electrons. So the loss of electrons from one substance is called oxidation. So how would you lose electrons? Electrons are lost by bonds being broken and shuffling around atoms plus their electrons, removing atoms and their electrons from one molecule and adding them to another. That's how electrons are lost. So when those electrons are lost, they have to be added to another molecule, right? Electrons represent energy, so we can't just lose energy. What we're going to do is transfer that energy. We're going to transfer those electrons. And so when we add electrons to another substance, we call that reduction. I know it kind of sounds counterintuitive, like you think, okay, we're going to add something to something else. How can that be a reduction? That doesn't make sense. Like if you think about the term, what the term reduction means, but I didn't make up the word. Somebody else did. But reduction is the addition of electrons from one substance to another substance. So when a molecule is oxidized, it loses one or more electrons, and then when it's reduced, it gains one or more electrons. So here's another example of energy coupling, or these reactions being coupled. You take away electrons from one substance or molecule, and then they must be added to another substance or molecule. So let me fix some of the formatting that we've got here, just so you guys don't get confused. I'm just going to rewrite the chemical formulas here. Okay, so we got the formatting fixed. Let's look at the reactants. You've got a molecule of glucose and 6 molecules of oxygen right here. If we follow the green arrows, if we compare what's happened to glucose on the reactant side versus the product side. What happens is glucose is oxidized. Glucose loses electrons in the form of hydrogen. So it's losing hydrogen atoms, and hydrogen atoms have electrons, right? So if we were just to, let's do this. Let's say, okay, well, if we lose hydrogens, we cross out those hydrogens, what do you have left? You have carbon and you have oxygen left. Well, look, that's what we have right here. So what's left, part of what's left when we oxidize glucose is carbon and oxygen. But those hydrogens that we take away from glucose have to go somewhere. Remember, this is energy, so energy is never lost or destroyed, it's just transformed. So we're just going to shuffle electrons or hydrogen atoms and their electrons around. So what happens is oxygen becomes reduced, or oxygen gets reduced. See this oxygen right here? Well, remember, reduction means adding hydrogens. So on the right side of the equation, here is that original oxygen, and what we've done is we've added hydrogens to it. So we say that oxygen becomes reduced. It has those hydrogens added to it. And so by breaking those hydrogen bonds, then we release that energy, and that energy goes towards creating ATP. How? Remember, in the cell, there would be existing adenosine diphosphate and free phosphate. Phosphate just kind of hanging around by itself. And the energy that comes from breaking those hydrogen bonds is used to add this phosphate to ADP, creating that high energy molecule that we're after called ATP. So that's an example of what happens to those hydrogen atoms and their electrons. Things are just getting shuffled around. Last week we learned about enzymes, and we learned that enzymes drive cellular reactions. That many cellular reactions would occur without enzymes, but they would occur so slow that they wouldn't be useful to the living organism. So we've got these specialized molecules that are usually proteins that speed up chemical reactions. They act as catalysts. They're going to lower the energy of activation so that chemical reactions can occur. And so in cellular respiration, one of the most important enzymes necessary to oxidize glucose, take away hydrogens from glucose, that's what oxidize means, right, is a molecule called NAD+. Really important in oxidizing glucose. It has the ability to accept electrons. So it accepts both electrons and the hydrogens. So it's a temporary carrier of electrons. When we remove the hydrogen atoms and the electrons from glucose, they temporarily have to be held on to by another molecule, by an enzyme, by NAD, or NAD plus rather. So NAD plus is going to be able to carry the hydrogen, carry its electrons, so it accepts electrons, and NAD as an enzyme then becomes reduced. Remember, reduced means the accepting of hydrogen atoms. So here at the beginning we call this NAD plus, and the plus just kind of means, hey, there's a spot on me to carry a hydrogen atom, and that's what happens. Let's have a look at what NAD looks like. I'm going to let me fix the formatting here real quick so you guys aren't confused. Hold on. Okay, so we got that formatting fixed. Let's look at let's just look at a generic reaction here of an oxidation-reduction reaction and how NAD plus works. So you have a here you have a 3-carbon molecule, and you've got some hydrogens right here, hydrogen bonds, anyways, here and here. So when a substance or molecule is oxidized, that means hydrogen bonds are broken. So if we look over on the right, we've lost these hydrogen bonds now. And so we get 2 free hydrogens. If you remember from the periodic table, hydrogen has 1 electron, right? Just has that 1 electron. So if we've lost 2 hydrogens, that means we also then have 2 electrons. So we've got hydrogen plus its 2 electrons. So if we look through the process, or we carry through that chemical reaction following the green arrow here, we would have NAD, this special enzyme existing inside the cell, and it has the ability to accept 1 hydrogen atom plus 2 electrons. Well, where does that happen? Well, the NAD can carry 2 electrons, so 1 here and 1 here, and it can accept only 1 hydrogen. So if we look to the other side of the equation here, to the products, what happens is NADH becomes reduced. It accepts hydrogen, 1 hydrogen atom itself, and this would be that hydrogen's electrons. But the thing about NAD is that it can carry 2 electrons. So it's going to carry another electron that belongs to that hydrogen. But it doesn't have room to carry this hydrogen itself. So this hydrogen is going to stay free in the cell. But the energy from that hydrogen, its electron, is actually carried by NADH. So we call, it's being carried by NAD in the form of NADH. So we call these electron carriers. NAD plus is an electron carrier. It's a carrier of energy. It's shuttling energy. And this is going to be temporary, right? All that NAD is doing is carrying those electrons to something else. Okay? And how that occurs. There are other electron carrier molecules that work very similarly to NAD+. There is FADH. We'll talk about a couple of the other enzymes used in cellular respiration that are similar to NAD. We'll talk about those a little bit later. But these carrier molecules that function like NAD, they essentially act as sort of individuals in a relay race, where one individual passes the baton on to another, and that person passes the baton on to another. These enzymes work very similarly. They form what scientists like to refer to as a staircase, and I'll show you a picture of that in a second. But if you have a bunch of these electron carriers ready to receive electrons, they basically make this chain of transferring energy. So the electrons pass from one carrier to the next, or down this, what we call this staircase. This is what we refer to as the electron transport chain. Think of it as a relay race, passing the baton on, passing the electrons on. And as those electrons are transported from one molecule from one molecule to the next down the chain, ATP is generated in the passing of these electrons, or the transferring of these electrons from one carrier to the next. The way that it is represented in your book is like this. So remember that NADH is carrying one hydrogen and two electrons, and that's the first carrier right here, would be the very first carrier. But what's going to happen is the two electrons are going to get dropped off, and they're going to get dropped off to the second carrier. Well, that's cool because all enzymes, as you know, are recyclable, right? So when those two electrons are dropped off, then we get NAD plus being recycled. Now it's got two spots to carry two additional electrons, so it can go back to the beginning of the chain and go through the process all over again. And this hydrogen right here that NADH was carrying, that hydrogen gets released just as a free hydrogen. So now we've got these electrons. Here I'll draw them in black. So you've got this carrier now holding two electrons. Well, that carrier is going to transfer the electrons next to the next carrier, number 3 in the chain. And when that happens, a little bit of energy is released. And then the third carrier is going to transport those electrons or pass those electrons off to number 4. And when that happens, a little bit of energy is released. And this continues on through what we call the electron transport chain. And ultimately, at some point, there isn't going to be another carrier to accept electrons. Not an enzyme anyways. So then what you get are 2 free electrons, which is bad. We can't have that, right? There has to be something, some terminal electron receptor, some final molecule in the chain that's going to accept those electrons, accept that energy. So when the enzymes have completely, or the electrons have been passed through all the enzymes, there has to be another molecule, something waiting to accept those electrons. Well, guess what? It is oxygen. Oxygen is waiting to accept those electrons. It is waiting to accept those hydrogens. And so as long as there is oxygen present, let me move this up. As long as there is oxygen present, what we end up with is H2O. We had this existing, let me write this for you, 1/2 O2. A half of an oxygen molecule means just 1 oxygen, right? So you had 1 oxygen atom waiting there to accept 2 hydrogens and its 2 electrons. So when we have 2 hydrogens and we add that to 1 oxygen, what do we get? Water. So we refer to, when we're learning about cellular respiration, we refer to oxygen as being the terminal electron receptor. That is the oxygen is going to grab onto that energy and make water out of it. This is why oxygen is required for most forms of cellular respiration. This is why you need to inhale. You need to get the oxygen into your blood, because that oxygen in your blood is acting as the terminal electron receptor. If there wasn't anything to accept these 2 electrons, those 2 bits of energy, you'd have free energy roaming around in the cell, which can be very, very damaging. The process just wouldn't work properly. So oxygen is absolutely essential for this process. This is why we call this cellular respiration, cellular breathing, because this molecule of oxygen, or the oxygen atom, is necessary as that terminal electron receptor. Now this is going to be the last slide, and I'm going to leave off on this slide because when we start with part 2 of the lecture, we'll start right up here. Now we've been talking about how this process of cellular respiration, how it occurs, or the basics of how it occurs, but we haven't yet really learned too much about where it occurs. I've said that this process occurs in the mitochondria, but let's take a closer look at the structure of mitochondria. Remember that relationship between structure and function. That something structure is going to tell us a lot about how it functions, or the function is a result of its structure. So if we look at the jelly bean shape of the mitochondrion in general, mitochondrion is singular, mitochondria is plural, you see that it looks like a jelly bean on the outside. It's got a membrane. What would that membrane be made of? You guys want to guess? What are all membranes made of? Phospholipid bilayer, right? So it's got this outer membrane, right? And we're pointing to the outer membrane here. So that's a phospholipid bilayer. And then just underneath that, there is what we call intermembrane space. So that intermembrane space is that space that you see right here. And then you see what's called an inner membrane. There's this snake-like structure that you see in green. And this entire snake-like structure that I'm just going to kind of begin to outline in black for you, this whole structure is called cristae. And that cristae, or that structure, is made, again, of a phospholipid bilayer. So when we're pointing to this structure, the green line here, and they're calling it that inner membrane, guess what? It's another membrane made of a phospholipid bilayer. And inside of there is going to be this space, right? And remember that membranes represent a barrier. And barriers represent a way to set up a concentration gradient. And concentration gradients then can turn potential energy into kinetic energy, the energy of movement. We'll talk more about this in the second part of the lecture, but I wanted to introduce the structure of mitochondria to you because cellular respiration is going to occur. Some parts of cellular respiration are going to occur outside of the mitochondria, here, like in the cytoplasm, but the majority of energy production occurs either within this inner membrane space here and over and through this inner membrane. So it's really important to understand the structure of that mitochondria. Okay, that's all I have for part 1. Stay tuned for part 2 coming up next.